All right. Thank you so much to our hosts and our participants for being here and making this happen. This is part of the ISPIC webinar series. This is a masterclass on the use of fermentation technology for protein production and especially for human nutrition applications. Um, I'm delighted to welcome our two panelists today, Professor K.V. Venkatesh, a professor of chemical engineering at IIT Bombay. He has been there for almost 3 decades, more than 3 decades. And we're very delighted to have you here and share your wisdom from all your years of experience. Professor Venkatesh has mentored dozens of PhD students, master's students over these 3 decades, and his lab work has extensively focused on the areas of systems biology, biochemical engineering, and food engineering. Besides being a full-time professor, Professor Venkatesh also is an entrepreneur, has founded MetFlux Research. It's an R&D company which has created an investigative physiology platform that uses genetic and physiological models integrated at the cellular tissue level, which functions as a tool with wide-ranging applications in clinical research analysis, including pharma and the food industry, food and nutraceutical industries. More recently, Professor Venkatesh has also been toe dipping in alt protein related entrepreneurship. He's going to be talking more about that at his presentation today. Thank you, Professor Venkatesh. Thank you so much for dedicating your time for this webinar session. Welcome. And secondly, I would like to invite my colleague from GFI US, Adam. He is the lead scientist in fermentation at GFI and has focused his career on the development of strains and process improvements for various fermentation platforms. At GFI, Adam's focus is mainly in biomass and precision fermentation biotechnology for food protein production. Adam's expertise, as I mentioned before, is also systems biology. And his goal throughout his career has been how do we better characterize these microbial systems and gain a deeper understanding of the whole bioprocess development, including strain engineering. Thank you, Adam. We can't wait to hear your insights into precision fermentation technology and how we can leverage that for food protein production. Thank you so much for joining us today. With that, I will invite Venkatesh sir to begin with your presentation. We're very excited.
Let me share. You're able to see my screen?
Yes, yes, we can.
You're able to see, right?
Correct. Yeah. Now it's in full. It's in presentation mode. It's great.
Okay. Thanks, Chandana, for that nice introduction. As Chandana was indicating, my core area is in systems biology. We have been modeling biological processes. If my video is off, it's okay. I will. Right. I'll give the presentation
totally
without
fine.
that. Yeah.
Totally fine. Yeah.
Some issues suddenly, this is scrubbed up. As Chandana was indicating, I work in the area of systems biology, essentially applying various analysis and mathematical tools in optimizing biological processes. And we have been doing microbial fermentation, fungal fermentation, and then quantifying how they grow, how to optimize their products. Because in nature, the organism itself is tuned for its survival, and it is not optimized for a product that we are as humans interested. Okay, they have their own evolutionary driving forces, and then as engineers, we need to manipulate it to optimize the biological processes for the needs that we have, right? And that is where some of the engineering techniques of systems biology and bioprocess optimization comes in. So today, what I'll talk about is the work that is going on in our lab, and then generally the issues that are there in especially protein production for human nutrition applications is what I'll try to address in the next 25 minutes. Three cell types that we work, and then which has also sort of shown promise in this application of protein production is obviously algal systems. Recently, we've been working on mushrooms as a protein source and how that can be used to optimize both in the mycelial and the fruit body systems, mushrooms, and obviously yeast and other microorganisms which can be used. So I'll sort of go through these examples as to how we are using algal, mushroom, and yeast systems to produce protein. And then I don't have to tell to this group that there is an immense growth potential for alternative protein market, and novel fermentation strategies will obviously help in achieving these goals, and in terms of producing a protein in the market by alternative methods. Obviously, it is a science intense applications that we have to look at. So we need to try to see what, how to improve yields, what are the culture combinations that will work, optimize the fermentation condition. And I think you'll hear more about precision fermentation that also comes into picture. And then obviously the use of the biomass, the feedstocks that need to go in. So we need to look at different aspects, and newer techniques like synthetic biology and the systems biology approaches will surely help in this achieving these goals that we have been talking about. Obviously, it is a challenging problem statement, and there are obstacles like consumer acceptance, economic viability, which can be brought about by nutritional levels, increased nutritional levels, growth on waste, so that the feedstock can be cheaper, enhanced flavors and odors, and then so these approaches need to be used. So to make this possible, some of the challenges that we need to address is in the economic viability of the feedstock choices that we need to make. Process design, reactor design and downstream processing optimization, where engineering principles have to be applied. Product characteristics, how do we get the same quality of products, each time we go through a processing? How do we eliminate the presence of undesired molecules which may come about? The complexity of food safety and protocols have to be addressed. And then ultimately, obviously, the consumer acceptance is the key. After we produce all those, and if there is a lower consumer acceptance, it becomes a challenge. So we need to, cross all these challenges before, it becomes a scalable and economical and a consumer acceptance, product. So some of the optimization strategy obviously is the, strain selections and improvement. And there are different ways of optimizing once you select an organism. So one of the ways is, you just do random mutagenesis, right? And then see whether you get strains which are better. And then in the past, this was extensively used. And even now, when we don't understand the organism's behavior very much. So, this is one of the approaches that is taken, that, you do random mutagenesis. So this is essentially linking the phenotype, is a forward genetics. That is, phenotype, you desire a phenotype, and then you randomly mutate a genotype and see whether, you are able to achieve that particular phenotype, right? And then you can also have a more directed approach where you have insertional mutations, where you sort of reverse genetics that you apply. And obviously there are various components to it in terms of time, cost, know-how, recovery, biosafety, and genotype. While the random mutagenesis is, maybe routinely done in labs with lesser costs, and, can keep on screening, but then, you're not very sure whether you're going to get the desired strain or not. Whereas a directed mutation is costly, but it'll give you very specific genotypes that you design, right? So in our lab, we are using both methods, trying to see where directed mutations can be used, or random mutagenesis is used. And the other approach in more directed is again adaptive laboratory evolution, where you try to get organisms which can grow better or can produce your product better by adaptive target pressures on the organism. And so you do a proper experimental design, give an adaptive target, and then try to get a strain which produces the product with a very high growth rate. That means that your productivities are very, very high, right? So again, this is one of the things that nowadays routinely being used. With a combination of all these three, we hope that we are going to achieve a strain which can be economically used in the process, in the bioprocess. And for the algal system that I give the example, we have gone through this process and then actually trying to optimize for faster growth rate. And then there are other techniques. If more knowledge is known about the organism, both at the sequence and the genome level, and the genes are annotated, one can build genome scale metabolic models. So this is again optimizing the metabolism or metabolic engineering that you carry out by modeling the genome a scale metabolic model for the organism, and then it will tell you where exactly you need to manipulate and which are the bottlenecks that exist towards the product or towards the growing of a particular organism, right? So for example, this was used to optimize the nitrate in the medium in one of the examples that we have, where we can actually try to see how genome scale models can be used to find out how much nitrate is needed and what is the uptake of nitrate that is needed towards higher growth rate and also towards a particular product. Okay, so such insights can be got by using genome scale models. And so this is optimizing at the strain level. So this is the basic information that we need, and it all depends on how much you understand about the strain. So a combination of experiments and analysis at multiple levels. So now you can have a lot of measurements at the omics level. You can get your RNA-seq data, metabolomics, and various measurements that you can carry out, which help in modeling the organism at the cellular level, and then try to optimize for a particular constituent, or try to manipulate its phenotype by manipulating the fluxes at various points in the system, right? So last 2 decades, approaches like this has been used more towards production of a particular protein or a particular product for pharma applications and others. And now we know it's also entering into producing huge quantity of proteins and in food applications. Right. And the final characteristic that you want to do, where the phenotype of the organism, yields the final product, where the product characteristic has to be, well characterized, and it has to be that you have a control on the product characteristics, right? That the product that you are going to produce, and that is where the precision fermentation would also come in, where you have a very controlled product outcomes that you get. Which may be difficult, for example, you grow algal in an open pond in the atmosphere, right? So each day you may have a variability in your products, right? So the product characteristics becomes very important, and then we want to see whether a specific product type is got. So for example, amino acid profiling itself, right, and how much of essential amino acids that you can get, right? So for example, if I compare beef to soya protein to protein from mushroom and protein from cholera, in the algal system, microalgal systems. So the last two are where we have from our own lab optimized and then got protein characteristics done. So you will see that mushrooms has close to 50% of the amino acids per 100 grams of the protein that you get from mushrooms are essential amino acids, whereas beef has about 43%. And you can see that the plant-based soya is only about 20%, right, of the essential amino acid. And the algal system is again somewhere in between, close to 31%. right? So the WHO standards are that your essential amino acid in any protein has to be, greater than about, I think 27 or 28% of, of the total protein. And in this case, you will see that beef, mushroom, and, the algal systems satisfy that WHO requirement of, how much of essential amino acids that are needed. Okay. And what we have been also trying to do is, can we do a combination of different strains of microalgal systems that we have, that we mix these proteins in such a way that, it is closer to beef, kind of composition. And in some instances, you want high lysine to be present, or in some instance, some other amino acid to be present. So can we play around with mixing different kinds of characteristics of protein to get, unique protein combination for very high-end therapeutic food applications as well, right? So some of those things can also be, attempted, right? So the product characteristics becomes very, very important, because that is the end goal that you want to achieve when you say that your product has these properties and these characteristics. Okay, so feedstock optimization. So this can be at some level, cost consideration that you need to go through as to which feedstock to use. Obviously in precision, and then you want a very precise, product end. The characteristics of the feedstock also, if it is very well benchmarked and characterized, then at the end you are going to get, a well characterized product itself, right? So gaseous feedstocks like CO2 and methane, right, are some of the options that exist, and we have been using these as well. And then obviously the wasteful biomass, right? Paddy grass, sawdust, especially in a solid state fermentation, where instead of the submerged fermentation, if you have a solid state fermentation, right? So you can have these feedstock that can be sort of used. And the other is obviously the bioprocess optimization, where the reactor design and the process control, trying to characterize the process itself that happens in the reactor, and how you can predict some of those based on the parameters that you use to characterize that bioprocess. And so how a reactor can be designed, and also how it can be controlled, so that you optimize at the process level. So you can start optimizing at various scales. So first you're optimizing at the cellular level, and then seeing how the cells themselves are able to produce a particular product. Okay. And then you say, what is the feedstock that I have to use, and how I can optimize that, in the reactor. And then you go and actually optimize the bioprocess itself, right? So the conditions that exist in the fermentation, and also the type of process that you want to use, right? So the bioprocess optimization also becomes important. And there are various types of reactors that exist, and it's all depending on the applications that you have, that you need to pick some of these. If it is your gaseous feedstock that you have, right? So you have a different design, and then if it is something else, mass transfer coefficient limiting for a particular substrate, so there are various considerations that are used in actually designing and then the choice of a reactor. But in many cases, bubble reactor is typically used. A tank and bubbling it and also controlled fermentation fermenters that are used to as a reactor, and conditions are optimized around that. And the final step before it goes into the market, obviously, is the downstream processing. So you need to harvest, dewater the the biomass, and then go through a series of downstream processing step, like sedimentation, flotation, flocculation, right? So various harvesting and dewatering processes that you need to go through, and in some cases additives may be required, although not desired, it may be required. So you try to avoid addition of other components when you do that, and then you have an extraction process. And again, here the choice depends on the product that you have, and then you need to optimize and try to see which of these truly work and which are scalable for the applications that you have. Enrichment, purification, and various processes that you need to use to downstream processing so that you optimize the yields of the final product and which match the characteristics that you desire becomes also sort of important. And in many instances, it's quite a in the whole process, the economics is quite high in the downstream processing as well. So I'll skip this. I mean, essentially what I had sort of indicated in the previous one, right? So you have to go through various steps to come to the final product. So I'll just give some, numbers, and some results that we have got in our labs. Although, as I told you, I was into systems biology and more at producing a compound, right? So how do you optimize lactic acid production? How do you optimize vitamin B12, right, through fermentation? So those were the applications that I went through. But in the last 5 to 6 years, we have been working more on trying to see how proteins can be produced. And so we have picked the algal and the fungal system. Towards sustainable food through algal and fungi. So essentially CO2, light, agro residues, and mineral salts. So those becomes our feedstock. And we want to have a controlled reaction. So we do an indoor cultivation of the algal and the fungi system. Fungi is essentially mushrooms. And then we go through the processing extractions, trying to characterize the product, the end product. For example, a mushroom protein extract or the algal protein extract, and characterize them very specifically. And while we are doing that, we also do get phytochemical extracts, beta glucan extracts, food colors as a byproduct, right? So when we process the proteins out of the algae and the fungal system, and then we do formulate in some instances towards functional food, wellness foods or nutraceutical supported foods and therapeutic foods, right? So a combination of the fungus and the algal protein, and also fermentation on those proteins themselves to get fermented foods, right? So some of those things we are trying to attempt. So go from agro residues, CO2, mineral salts to functional foods and protein formulations, right? So that is the approach that we have been taking. So towards this, we have got a chlorella species, right, isolated from northeast of India, which is very unique, because the strain that we have is able to produce huge amount of protein. Typically algal systems have a tendency to store, fat. So this is one of the strains, which is a freshwater chlorella species, which produces close to 60% protein. And the characteristic I had shown, which is somewhere between, about 31% of essential amino acids that these proteins contain, which is higher than what the WHO requires for, satisfying the essential amino acid as a protein source, correct? So it has about 6% chlorophyll. So this is the uniqueness of this strain, that it has a higher amount of chlorophyll and a higher amount of protein that is typically seen in the algal systems, and about 3 to 4% fats and about 20 to 25% of carbohydrate. Okay, so this can become a rich source of protein. So because it has got 16% protein. And we also dabble with other strains. This is more for the EPA production and, so this is again algal system, Dunaliella salina system, right? And here, icosapentaenoic acid, which is a nutraceutical. So that we are trying to see how we can optimize, using this particular strain. Okay, so algal systems is one of the things that we do work in the lab, and for the particular chlorella species that I was talking about. So obviously, optimization of the light, the CO2, and the other micronutrients. So this is becomes a feedstock. And so we have optimized and now have a patented process of both the reactor design and the constituents to produce the 60% protein, right? Using a photobioreactor, which is again a very specialized reactor that we need to design, and how the light distribution has to be optimized is some of the things that we have worked, where again these proteins are optimized so that the same type of protein, at the end, we achieve. Correct. So again, here it is a combination of the approaches that I was telling you, optimization at the cellular level, then at the process level, process design. So at multiple levels, we do optimize, and we also use genome scale models and then things like that for trying to manipulate the metabolism in these strains. So using this, we have been able to extract food colorant from the algal systems, a nutraceutical grade chlorophyll, and chlorophyll, we process that nutraceutical grade chlorophyll to food colorants, and then obviously also produce this protein extract, and then some amino acid extracts also. So we can do hydrolysis of this protein to get some amino acid extracts. So, so from a algal strain, which uniquely from the Northeast India that we have got, we have been able to, convert water, CO2, light, and some nutrients to nutraceuticals, food color, and a protein-rich superfood. Right? And the other algal systems we are using to get EPA and DHA. Right? So just to give you, so these become the primary product. So although we get a protein-rich superfood, we also get chlorophyll in this case as a side product, which is of very high value. Okay. So we have calculated and optimized the optimized process that we have shows the advantages that you have. For example, producing protein from pulses, right? So in India, we use a lot of pulses as a protein source. Many of us being vegetarians, most of our protein comes from the pulses, right? So the protein yield from this process, for example, using CO2 is 1,000 times higher than that for the pulses in terms of total yield, right? Whereas the algal protein is producing about close to 53 kg per meter square per year, whereas the pulse protein, right, if you actually grow and harvest the pulses, so that is only about 0.05 kg per meter square per year. You can see the huge difference in the the yields and the productivity that you can get, okay, using such processes. Obviously, this also, because we are taking CO2 as a raw material, the carbon footprint also sort of reduces. So the carbon fixation is 300 times most faster than a fastest growing forest, for example. Correct. So 1 kg algal biomass fixes about 420 grams CO2 per meter square per day against 1.3 grams per meter square day from a forest. Okay, and this can also be improved further. And in addition to that, what we have seen is there are social benefits of optimizing, because this does use water. So the water can be harvested, and then there is a rural employment generations that you have if it is placed positively. So these are other advantages that you can add on, combining with this process. So just to give some mass balance, right? So for example, if you take about 1 kg of CO2, right? So we can, so they've slightly gone higher than this. So about 0.6 kg biomass, and then you can get all these products and close to 0.45 kg super rich protein supplements that you can get. Okay. So if it's just per kg CO2 cost that you see in terms of the this one, so you have a huge value add that you get by fixing this carbon dioxide using this algal system and the optimized process that we have developed. And then we have now gone ahead to try to see whether we can produce it in the tons, per annum. And then we have calculated, and then we have already now scaling up and trying to see whether we can achieve, at scale, trying to do. So it is not always possible when you scale. Scaling is another challenge of these biological processes, because what you see in a shake flask and what you see in a 5-liter fermenter and what you see in a, in a 500-liter or 5,000-liter. So the phenotype changes, right? So because of the organism has to respond to its environment, and it all depends on the scale that you're doing, how their phenotypic response is when you scale up. So in many instances, it is not possible that you can scale to a very high level. So in such cases, for example, the algal system we have seen that about 150 kiloliters, right? So beyond that, it's very difficult to replicate. So if you want a scale, like for example, 200 tons per annum, right? So then you need to use multiple reactors, right? So you replicate the reactor at a smaller volume and then create the same thing, and that is more optimal than using a huge reactor, right? So we've been trying to optimize how we can produce this protein using algal systems and how they can be optimally scaled. So some of these things we are attempting at IIT. So another example is, again, here, a genome scale model was used to convert methane to a single cell protein, right? So again, you do a lot of metabolic engineering and try to use methanotropes, right, to try to see how methane can be used. And so we helped one of the companies also, we worked with them. So there is a company called String Bio in Bangalore, which uses methane to produce single cell proteins. And so now the protein that they're producing is more for animal feed, but they also have the aim of trying to get human consumption protein as well. So again, from biogas, you get your methane, and then you use that to try to see whether you can produce a single cell protein. So this is another example where you use a gas, a biogas stream to convert that into a protein. So these examples do exist, and now more and more such success stories are coming out. Okay. The other place that we have been using as a protein source is mushrooms, right? So one of the things that mushrooms, you can grow them, because it is a solid state fermentation. If you are looking for a fruit body, and if you are trying to do a solid state fermentation, it is very scalable, right? So space is the only requirement that is there. Otherwise, you can scale this to any extent that you want. Right? So we have been, trying to optimize, and again, use the same, cellular optimization, then feedstock optimization, how the product can be very, very specific. So we have taken the same approach, a scientific approach, from step to step towards optimizing a product. So you will see many people growing it just as a hobby. Right, so they take some solid waste and they put mushroom, mycelium, and then they grow. And there is a protocol, so they follow the protocol, and then you get your fruit body, right? So it is, many times it is by chance. So many small players do that. I know they grow different types of mushrooms, even in India. Although China and Malaysia and all that are the leaders in these mushroom production. Right? So what we have done is we have taken a very scientific approach, so that at the end we need to have the same composition, right? So in an organic sense, we have grown these mushrooms and trying to extract protein from the mushroom growth. So obviously, mushroom have an added advantage of they are having a lot of nutraceuticals, right? The ergothionein, gluran, beta glucan, lectins. So along with your protein, if you're able to add this, so you have an additional advantage of a protein source with, antioxidants and, health benefits that you get, which are including micronutrients. And it has been used for various disease, right, as antiviral, antioxidants. There are a lot of studies, clinical studies as well, showing the benefits of some of these nutraceuticals that the mushroom produces. So again, manipulating these micronutrients and nutraceuticals along with the protein that is there, correct? So gives this added advantage of using such, for example, oyster mushrooms that we have sort of optimized. Okay. So again, here the optimization at both levels. So we do solid state fermentation, which is again using agro waste, right? So it could be sawdust, it could be, for example, the jackfruit skin on the top, jackfruit skin. So each one gives unique combinations of characteristics of the mushrooms that is grown on that. And then we also do mycelial of the same mushrooms with a submerged fermentation where we optimize for the mycelium, right? So the protein can come from both the sources, from a more precision fermentation kind of process or a solid state fermentation process, right? And both are sort of scalable in terms of a desired product characteristics that we want. So we have been doing this, few years, trying to optimize these processes. And then again, as I said, the product characteristics become very, very important. In this, we want a precise, micronutrients, vitamins that at the end of the, the growth and separations that we do. So we need to have a control on that, right? If each day, just like any other harvesting, if you grow mushroom, so you will have different composition on different days, but we want a more controlled, outputs is what we are trying to attempt. And even here, we have seen that some of the oyster mushrooms have very high protein content, right? I don't know whether which one is saying. For example, the Dejamur has quite high. Now we have gone almost to 42% protein, in the Dejamur, right? And then again, the P. ostreatus, again, this has gone to about 36. The way that we have sort of optimized the feedstock and the growth of some of these organisms. So again, can we have some of the future things that we're looking at is can we have a combinatorial nutritional products with algal and the fungal system to be mixed with the nutraceuticals that are present. Use of extracted biomass as a feed material for other fermentation. Mycelial production, can we scale that purely as grow it as a mycelia and then not convert that into a fruit body so that it is more controlled. So some of these things, we are sort of attempting in our lab. So I think I'll stop here. I hope I've taken that 30 minutes that was given to me. Yeah. Great. I'll stop here. And I think Adam can take over, or it's a question of,
we have received a few questions, Professor, and I think first of all, thank you so much for that super informative presentation. I learned so much, so thank you for that. The questions are coming in. Let's give it some more time. Maybe we can hear Adam's presentation and then take questions all together in the end. Is that okay?
Yeah, absolutely. That should be fine. Yeah.
Thank you. And now Adam, we're very excited to hear what you have on precision fermentation and all proteins there. Take it away.
Great, thanks so much. I will pull up my screen and begin my slideshow. Chandana, can you see this okay?
Yes, we can.
Excellent. Yeah. So first of all, good evening and welcome. And thanks for Chandana and GFI India to get this together. This looks like a great program for ISPIC and for getting everybody together to really push forward on an industry that is growing and exciting and has a lot of space for innovators and truly has, I think, a great potential to move everything forward in terms of sustainability. So I'll go through a really quick outline. I don't have to give you an introduction to GFI, but I just want to show you the little map to show how interconnected our teams are. I think our overview of fermentation was excellent from Professor Venkatesh, so I can skim through that, and then I can get a little bit into what fermentation and particularly precision fermentation looks like now, and how progress moving forward is going to accelerate because of new tools that we have, especially on the strain development and bioprocess side, and then speak about some of the challenges that may need to be overcome. And that's where there may be, even more space for innovation here. So GFI is now, this number continues to change in the bottom, but it says over 170, I believe it's over 180 staff worldwide in 6 regions. So I work in our science and technology pillar, and I work here in the US. I'm based in North Carolina, and I'm our lead scientist in fermentation. So I work with a team of another fermentation scientist, and he's a former bioprocess scientist from industry. I'm really what I did was transcriptomics and data analysis to look at process development, strain development. So there are a lot of those tools that you can use upstream, like looking at random mutagenesis or trying to knock out genes or overexpress genes. One of the things you have to do is try to observe what the effects are there and then see how you might be able to use those to inform the others. So that's what I did. I'm here now at GFI for almost a year, and we're a non-for-profit 501c3, and we're developing the roadmap for alt proteins and a sustainable future. So we need to do this sustainably, efficiently, and safely. And that's really exciting because I think fermentation can do something that a lot of our other platforms potentially haven't unlocked yet. But you can see that in a relatively compact amount of space, we're able to take fermentation and make a high density of product. So if we're looking at our current worldwide agricultural lands, some 77% of that goes to livestock or making feed for livestock. But really worldwide, only about a third of the protein comes from meat and dairy. So GFI's approach obviously is to use plant-based, fermentation-based, and potentially cultivated meat-based as these become available in different regions to alleviate this stress on our environment, but also pave the path forward for when our worldwide population approaches 10 billion people in 2050. So I'm going to give you a quick overview of the fermentation landscape, especially as it pertains to precision fermentation. So like Professor Venkatesh told us before, fermentation, if you look at the metabolic definition, is a little bit different than the definition we use here in food and an alternative protein. But really, it's the cultivation of microorganisms to produce a bioproduct for food. So we're looking at the potential to use bacteria, yeast form fungi, filamentous or mycelial fungi, microalgae, and potentially other microbes such as archaea. We've been doing this for a very long time. Microbes have been a part of our worldwide human food system almost since the beginning of culture. And that's because they're able to provide us with micronutrients, with stabilizing compounds that allow for the preservation of foods, or they do things like dramatically change the pH of our foods to allow for these foods to have longer shelf lives and things like that. But for the past 100 or so years, we've been using microbes in our biomanufacturing processes to do things that otherwise we would have to extract in a more intensive manner from something like plants. And I'll go into that a little bit in a little while. But also, as anybody who may have come from biotech or from pharma has seen, we can use microbes to make things like vaccine peptides or biologic drugs that can be used in the treatment of a variety of conditions. So when we look at fermentation for alternative proteins, there's traditional fermentation has been used for 10,000 years or potentially more. Biomass fermentation, which I think had an excellent overview from Professor Venkatesh. And then a relatively new innovation on the market that has come in, which is precision fermentation. So precision fermentation is using microbial hosts to produce specific ingredients that often require greater purity, but also require relatively large quantities of that particular ingredient to be made by the microbe. So in foods, we're often thinking about proteins, but we can also think about fats or other ingredients like volatile compounds or pigments. And these are ingredients that can enable improved sensory characteristics and functional attributes, especially for things like plant-based proteins, or even potentially in the future for cultivated meat. So the landscape of fermentation-derived proteins, I told you fermentation, precision fermentation requires a bit more of labor because it requires higher purity. We're not necessarily just taking that biomass and treating it maybe to reduce the amount of nucleic acid or to separate it into its groupings of fats and carbohydrates and proteins, but we're really looking for one particular ingredient or one particular class of ingredients. And we have been doing this for a while. Like I alluded to before, enzymes have been produced by precision fermentation for a long time. Often we use microbes and we look at their natural repertoire of enzymes that they create, and we grow them up to get those enzymes and then use them industrially. So enzymes like amylases, which break down starch, cellulases, which break down cellulose, are endogenous or sort of native to those microbes. And we have developed lots of different ways to grow up those microbes to produce lots of those enzymes and then process those away during downstream processing after the upstream processing or fermentation will. We're starting to do things like make growth factors in fermentation, and these growth factors can be fed back into our cultivated meat space, where instead of taking animal-derived growth factors, we can use growth factors that activate those cells and help to differentiate them into the proper cell types to make cultivated meat. And we've also been doing this on the amino acid and vitamin side for a long time. Microbially produced amino acids are a mainstay of the animal feed industry, and continue to do so. And so we'll be probably seeing this. I saw a question before for the basal media components for cultivated meat. Many microbes can produce large amounts of particular amino acids that can be then downstream processed from fermentation. So when we think about precision fermentation, there really are two different types, and we see companies using these two different types for lots of products. So there are organisms that have been bioprospected and selected for native traits and ingredients. And some of the so-called easier ones to look at are if you take maybe a fungi, and that fungi has a distinct red pigment, then that red pigment might be something that you would like to take and grow that fungi in a reactor, and then isolate that red pigment away to get a natural red food pigment. Other times, there's engineered genes that go into these microbes. So you may be interested in an egg protein, so egg white protein like ovalbumin from chicken eggs, or potentially ovalbumin from other bird species that might have different properties. You can take that gene, adapt that gene so that it is read more effectively by a yeast-based fungi or a filamentous fungi, introduce that gene along with the machine, with the genetic components that the expression machinery need to read that gene, and then try to produce as much of that as possible. So companies now are using these to make dairy proteins or to make egg proteins, and try to produce as much as they can in a way that would make it as easy as possible to bioprocess that particular protein away from the rest of the biomass. So if we're looking at that bioprocess, we can start with a gene, potentially a gene from an egg, from chicken egg, that would allow for ovalbumin, the one particular protein in egg whites, to be produced, and introduce that into the production organism. And after lots of R&D cycles, find the right strain candidate which matches all of those genetic components with the right location in the genome, with all of the other improvements you may have made to the strain, and finally get that strain to produce it at a level that could be commercially viable. And then that must be put into our upstream processing to grow the microbe up and also have it produce that particular protein. And then you have a waste stream of the spent media, the production organism, all of the nucleic acid, and purified protein. And we can talk about the purification process in a little while. And often in precision fermentation, we're thinking about proteins that have already existed in food systems. So egg white proteins are used by bakeries or used by other food formulators and consumer product good companies to produce lots of products, snack products or things like that. And there has been a push to make these in animal-free ways. So a lot of these proteins have come into our food systems because they're useful for formulation. They might help with foaming. They might help with formulation to help keep things really sticky. For cheeses, things like casein proteins, which comes from mammal milk, is a protein that helps really get stretch. And once you get this stretch, you have that familiar bite that you can get from cheese. And many times plant-based cheeses can't replicate that yet. So these are proteins that are meant to improve the processes, but also improve the products. And many times they can be used as sort of drop-in replacements for proteins that are already in there. So if we look at biomass fermentation or precision fermentation, often they're quite similar in that a microorganism needs to be added to its media to interact, and you need to use that feedstock so that it can produce the proper amount of biomass and then produce the particular product. And when we're thinking about media for precision fermentation, I would have to say we're not quite as innovative yet in that space as we are in the biomass space. Many of the companies making precision fermentation products still rely heavily on highly purified, not quite pharma grade, but sort of things like food grade glucose or dextrose equivalent syrups, and then purified nitrogen sources to grow up their organisms. So a real innovation here could be using readily available feedstocks that could go into the precision fermentation realm, but also provide the same kind of nutrition at the fermentation durations that people would like. You could give a lot of these organisms more complicated starches or ag waste streams. However, their fermentation times would be so much longer, it would become economically inviable for them to do so currently. We're hoping that that changes in the future. So when we get to precision fermentation, there is more on the protein isolation side, and I'm going to talk about that in a little while. So when we look at biomass, so if we take a process like corn, which is a mycelial microprotein that originated with the Marlowe Foods Company in the UK, they use a mycelium, and that mycelium is that underground root network below the fruiting body, and they use a fungi that has a really small fruiting body, but they can also influence it to grow in a way in liquid that fruiting bodies aren't made. So you just get this huge mass of mycelium. They have a relatively straightforward downstream bioprocess that they've been able to share because they've been doing it for long enough that it's off patent now. And part of it is dewatering and clarifying their process, which they harvest off of continuous runs of up to about 1,000 hours. Then they heat their biomass, which they call a mycoprotein dough. And this heating helps to remove the nucleic acids that if you left them in would cause gout. And then they freeze dry it and they freeze fractionate it, which helps to structure the proteins. And then they dry it and formulate it. In precision fermentation, when we're looking for a particular protein, we may have to do something where we take the media. So if this is a precision fermentation product that is made in the cell, but then trafficked into the extracellular space, we may take all of that extracellular liquid that has to be dewatered and clarified. So the mass and all of the particulates and proteins and debris that are in that mass must be concentrated. And then often it's pH adjusted and filtered so that you can get closer and closer to the size that you would like and to the sort of right protein folding that you would need for your particular protein. And then these proteins can go through a series of steps of concentration and diafiltration, which allow for the charge properties and the size of the protein to let it be purified away from all of the rest of that mass. And then finally, it can be dried. And often these are sold as protein powders or high concentrated slurries that go into other foods. So again, this is just a recap of our landscape of fermentation derived products. We're now seeing this space right here in precision fermentation, where we don't need something that's quite as pure or folded potentially as enzymes, and we don't need something that's maybe pharma grade or biotech grade like growth factors, but we're seeing this space right here really come alive for proteins. Companies are making use of this, and I think one of the really interesting ways to talk about this industry is to talk about the things that people are doing now and really appreciate the diversity of approaches and the diversity of products that are being made. And I think one of the things that I'll say is, if you have an idea and you see another company is making that same particular protein, they're likely using a different microbe, a different bioprocess, a different feedstock, or potentially a completely different strain innovation than you might be thinking of. There will be lots of space for many different innovations here to take over different niches that all allow us to sort of drive the sustainability and lower cost of this industry. So biomass fermentation is I think where the innovation started. Marlowe Foods really commercialized corn in starting in the 1960s with the R&D efforts, and then to the late 80s. So it took 20 some years to get to a commercial bioprocess for their product and to get to the right regulatory state. These things are happening much faster now. And I think there are companies like Solar Foods, Air Protein. These are companies that are using some of these gas fermentation systems. And Solar Foods has now in Singapore is making available their Solein protein powder, which is made from this bioprocess where they're capturing carbon gas and then turning it into a bulk bulk protein from single cell protein. But precision fermentation can also address some of these really specific needs that food systems have, and then give us the opportunity to move away from animal sources to make proteins that people have used in food systems. So I used egg white proteins as an example. Milk proteins like casein, whey, and lactoferrin, are all across the value chain when it comes to the particular products that you can make. So lactoferrin is a really interesting iron binding or ferrin protein that people are recognizing its potential use in nutritional supplements as an immune modulator and also potentially a bioprotectant. It's also a component of human breast milk as well. So there are companies that are trying to make baby formulas that have these proteins that otherwise aren't captured in baby formulations now. So lactoferrin could be added in, which helps to make a more nutritious baby formula. Whey protein is sort of a bulk item from milk, and whey protein can be added to things like ice creams and make really creamy and smooth ice creams. And you can get these types of flavors and mouthfeels in a way that you can't necessarily do unless you add animal-free whey proteins. And then casein, again, I think this is something that a lot of companies are pushing for, because plant-based cheeses have so far proven to be somewhat unsatisfactory on taste and texture, and caseins really provide the taste, texture, and mouthfeel of cheeses that people want and come to recognize from cheese products. Collagens and gelatins and proteins like that are relatively simple, straightforward proteins, but are used in many different places. So from cosmetics to things like candies, collagen helps as a really good binder. And what we're seeing now are companies that are using bacterial production to make collagens, and then using these to make animal-free versions of some of these places where otherwise people wouldn't always suspect that there's an animal protein in there. Heme protein has proven to be a hot topic. And so here in the US, Impossible Foods, when they made their plant-based burgers, they decided to add an ingredient that would include an iron binding protein that gave the color, taste of iron, and sort of some of the aromas during cooking that people associate with animal meat. And so they used a soy root protein that they were able to make in a yeast fungi called Comicadella pfaffii or Pichia pastoris, and then use that protein as an ingredient. And it's maybe only 1% of the weight to volume of their plant-based meat, but it's proven to be an exciting innovation for them. And there are other companies that are currently innovating on other heme proteins, like myoglobin particularly, which is the muscle heme protein, but others as well. And then finally, I spoke a little bit about growth factors. I think this is a way that a lot of folks in cultivated meat are thinking about using animal-free media formulations, because most people that want cultivated meat don't necessarily want the ingredients that go into growing it to be animal-derived. So growth factors, I think, are going to be a relatively high value target for precision fermentation. And this just gives us an overview of all of the different ways people are using these ingredients to formulate different products. So some of these products, like aquacultured foods is a good example, are, it seems like, using an approach where they're using more than one microbe, where one microbe might make a flavor compound that might make something more of like a seafood meaty flavor. And then they may also have one that covers sort of omega-3 fatty acid production, or something that provides more texture. But like I alluded to before, it's not just animal proteins that people think about, but things like pet foods. Pet food is a place where actually here in the US, domestic and companion animals make up about a third of the protein consumption of our country. So a really easy place to start to make an impact would be in pet foods. So Bond Pet Foods has a yeast product where they're making particular compounds that taste to a pet like chicken. So it gives them that flavor. And then they have a yeast-based dog food. Honey is another place. So Melly Bio is using fermentation to produce honey. And fats and oils, I think, are a real key to taste. A lot of times when we're looking at foods, we're smelling those foods or tasting them. Fat really gives us a lot of the flavor. Fat also gives us a really good culinary opportunity to cook something at a low temperature, at a high temperature, and do the things that we want to do with the rest of the food as well. And there are companies that are making alternative fats by fermentation. So there are companies like Nourish that are taking what we don't have complete insight to all of their processes, but they're taking microbial fats and then using particular downstream processing to get the mix of microbial fats that best replicate animal fats in cooking. C16 Biosciences is using recombinant pathways to make palm oil. So palm oil is an effective cooking oil. We can get it at really high yields from palm. However, the replacement of rainforest with palm plantations means that palm is not a sustainable oil for our environment and eventually for our economy as we see biodiversity go away. So C16 is using microbes to make palm oils. And now we've seen C16 commercialize their first product. It's in cosmetics first, but we're, I think, going to see C16 making fat and oil for human consumption once they are able to scale to the volumes necessary to make these products. And then finally, seafood. Much of the nutritional value of seafood actually comes from lower in the food chain, and omega-3 fatty acids that fish consume and bioaccumulate, which really comes from lower down in the food chain, from the microalgae. So there are lots of companies interested in using fermentation to get omega-3 fatty acids. I can skip this slide, but there's a lot of companies that are, one, interested in food ingredients, and two, interested in industrial fermentation that are getting involved in this space. And I think this is a great sort of intersection between companies that have expertise in food formulation to say, those are the proteins we want, yes, but if you're going to make us a casein, we need this casein at this pH or this moisture level, or potentially, something that can be cleaved by chymotrypsin to make the curds and whey when you're making cheese, or potentially something that acts like it had already been done so. And a lot of the companies on this slide have enormous fermentation capabilities and capacities. So seeing companies like this that have expertise and capacity in fermentation is really exciting, because one of the things that we're going to need going forward are the stainless steel tanks and the downstream processing equipment to actually produce these proteins. So why now? Why is this happening now? And I think Professor Venkatesh gave a good introduction to a lot of the new enabling technologies in biotechnology that we've been able to use. And I think one of the cool things when I look at this slide is whenever we have a really good innovation in biomanufacturing and fermentation, that innovation helps fermentation take over a space. So in the 1920s, people figured out that you could use bacteria to make citric acid rather than pressing it from citrus rinds. And within 10 years, in the 1920s, that completely took over the industry. If you look at insulin, when we were able to not take insulin from bovine pancreas and instead use recombinant protein made in yeast that's able to make those right post-translational modifications to get insulin folded properly, non-animal insulin took over all of that industry. And the same thing happened for rennet for cheese making. And rennet was really the first example of in the US where the FDA approved a recombinant protein that could be used in food system, in this case, a processing enzyme. So fermentation is poised for rapid growth. We can see fermenters, if you've ever driven by a 600,000 liter or 1 million liter fermenter for bioethanol, they're enormous. The potential to make a lot of product in one place is very large. It can be relatively low cost. It's familiar to the food systems because we've been using fermentation for a long time. And compared to plant-based or cultivated meat innovations, R&D cycles and production cycles can be really, really fast. Some of the filamentous fungi that are being used, particularly in biomass fermentation, like Neurospora crassa, you can actually watch Neurospora grow. It grows so fast. And it's sustainable, and we're making it more sustainable. There's a lot of energy inputs. If we can use clean energy inputs, if we can do a really good job of circulating wastewater in a way that makes it most efficient within a facility, and potentially if we can increase the yields, we can make this even more sustainable. We're now sitting on a world where a lot of the systems biology approaches that I hoped would happen when I was sitting in a systems biology lab are indeed occurring. And I think there are also other innovations along the value chain that are helping out here. So one is that we can sequence almost anything we want to cheaply and quickly. We can get a functional protein from, say, an animal and know the sequence of that the next week, and then start to synthesize a codon optimized version of that for a microbe. big data. I think one of the interesting things that we are just starting to get interested in the alt protein space is we think a lot about animal proteins and animal protein drop-ins. But if you want a foaming protein, does that foaming protein need to be an animal foaming protein? Or potentially could you find something from the plant world or even a microbe that contains a protein that might make really good foam? Anyone who's done fermentations that get mixed by an impeller will see that there's definitely some good foaming that can happen with a microbe. And we can use structural prediction and functional prediction algorithms like AlphaFold and AlphaFold2 to get at what some of the functionality of different proteins might be across all the kingdoms of nature and figure out maybe there are other proteins that would fit really well within our food systems. We can do faster and more informed R&D cycles. And I think on here in this slide, you can see, there are ways to network and use lots of small bioreactors to use those bioreactors in a way where maybe we can pressurize them to the level of an industrial bioreactor, or we can change the pH or build out different stress parameters that allow us to figure out what parts of bioprocess are going to work before we start to scale up. And using this kind of these informed cycles, we can do what's called design, build, test, learn to keep iterating on the same idea to continuously improve it. And at that point, we can go from a flask to a bioreactor that is a 1.5 liter bioreactor on a benchtop to a scale up fermenter, and then finally to production more quickly than we ever have been able to. So that road to commercialization is a little bit complicated. And one of the things that we're challenged with in alternative proteins for foods is to make foods economically. So a lot of biomanufacturing has come from pharma, and a lot of biomanufacturing has come from commodity level chemicals or fuels. So when we look at pharma, really, especially in the US, the sky's the limit really for what could be charged for many of these biologic drugs that are made by fermentation. On the same end, there's a lot of actually really cheap and effective fermentations that happen on the fuel side. So in the US, at least 10% of all fuel is ethanol, mixed with ethanol with gasoline. Sometimes that's up to 85%, depending on the car and depending on the regional demands. And so we can see that these can be cost competitive with what's actually out there for petroleum-based fuels. Food needs to be more pure than ethanol for fuel, and food needs to be less costly than a chemotherapeutic drug. And really, a lot of our processing technologies and bioreactor technologies have come from one or the other of these. So the space still needs more innovation to get effective downstream processing and separations, but without the energy intensivity and the relatively low value inputs of pharma. But it also needs to be able to get to this sort of the scales that we're looking at for something like fuel. We are seeing that happen. So costs are coming down for fermentation because there's something of a predictable curve in terms of the innovation space that's happening. So if we look at this Rethink X figure that came out, I think 2 years ago, maybe 3 years ago now, we're on a cost curve trajectory where we're going to get many of these proteins down to $10 a kilogram, which for something like a casein protein for cheese is going to be economically viable. For something like egg white protein, it's going to be economically viable. So this threshold will really enable many of the bioprocesses that we're looking at to be economically viable. But we have to think about that bioprocess. And so when you're looking at something that you may start on the benchtop to going to something that's going to be a full-on 600,000 liter 4-fermenter bioprocess, this is going to be a multivariable problem. So there's a lot of new tools out there that are really geared at helping to do some of the math. So scalar.bio is a new techno-economic analysis tool that tries to identify the costs of building a facility, the cost of feedstock inputs, the efficiency of the particular organism you're looking at, and then the downstream processing efficiency to say, hey, where are my cutoff points for making a viable commercial bioprocess? And it does have a lot of parts. Financing as a part of this, but feedstock and media optimization. Can you use the minimum amount of carbon inputs or nitrogen inputs that would make it so that your organism grows effectively? Can you invest in strain development enough to get a strain that doesn't make side products that are taking away from what you're making? Then finally, can you get the titer of your downstream processing to be high enough so you're not leaving some of your valuable protein caked on filtration systems somewhere within your bioprocess. And then lastly, when at the end of the day, what's coming out? Do you have a target that looks like the target that you intend to make? Does it replace whey protein powder in the way that you want to replace whey protein powder? So this is one of these figures that I look at and think there's so many moving parts to fermentation. And then when you look at precision fermentation, it's not just one of these graphs, but it's a graph for Pichia yeast making a dairy protein. It's a graph for Pichia yeast making egg protein. It's a graph for Trichoderma ricei filamentous fungi making one of these proteins, or potentially making a protein from a plant root that helps to foam. So there's a lot in this space that's left to do, and that's what makes it really exciting, because it is quite new, and we can use this enormous repertoire of biomanufacturing technology from fermentation to make this happen. So again, strain development, feedstock and waste valorization, and improved downstream processes are, I think, the really big innovation priorities that we can make here. If you have a grand idea, an idea for something that goes from ideation all the way to commercial bioprocess, if you can innovate along these 3 steps within it, I think that you're going to help save yourself money and drive that idea forward. And finally, this is something that Chandana and I were speaking about a little bit before this. We need more fermentation facilities. A key barrier is really using the right resources to build up pilot and demonstration scale facilities, and then to unlock all of the commercial capacity that could be made, but also that currently exists. And there are nations that are poised to do this because they have a lot of fermentation capacity. And that's an exciting thing about the Indian bioeconomy as well. And those bioprocess challenges, I think these are a great place for training. Whenever you have a pilot or demonstration scale, often these happen at a contract institution or potentially at a research site. And these are places where really a lot of expertise is learned, but it is gained as well by people training in those spaces, then potentially moving on to some of these companies. And last, I'll just tell you, I think that these are wonderful and they're a great resource. So if you're looking for more information about the current state of, in some ways, research, in some ways, what's happening on the commercial side in terms of what companies are making what, or who's investing in what, these are great documents. So go take a look at the fermentation state of the industry from last year that we released in April. And we'll have another one again next April for 2023, and then hopefully in the next few years we'll see something coming out of this program and see it in the future state of the industry reports. So thanks for your time. I know I didn't leave too much time for questions, but let's get right to that, and hopefully we can answer them quickly. Thank you.
Thank you, Adam. Thank you. This field is so fascinating. I could listen to this all day. The potential is just brilliant just to think about it. Like you said, it's very exciting. And I hope, by the way, speaking of state of the industry reports, GFI India is coming out with its own state of the industry, just talking about the Indian ecosystem. And to the point you just made, I think there are a few companies that we talk about there, which are spin-offs from ISPIC. So I hope that number grows, like you said, over the years, and we see more innovation coming out of this program that can really make it to scale. That said, I guess we can open up to questions. Please, if you have anything, please continue to type it in the questions box. Let me start. I think there are a few here. Question for Professor Venkatesh. Are you not using bioavailability markers like PDCAS for evaluating protein quality? Oh, I guess this is when you showed the table about comparing the protein content and the various amino acid content from beef and the various. I think they're asking why aren't you using just a PDCAS number calculating that protein quality? I suppose it can be done. It's not something that can't be done, right, Professor Venkatesh? I think you showed a more granular look at the different amino acid distributions. This is what I thought when it comes to essential amino acid distribution.
Yeah, I was muted. So Yeah. So you are right. We have not put any markers, but because we have, actually get the amino acid distribution we get. So we actually go and measure that straight away, digest it and then get the complete amino acid levels, right? And that's how we've been doing.
Perfect. That's good enough. Can we use symbiosis partnership between algae and fungi for more protein content? Professor Venkatesh, you're perfectly posed to answer this because you are pursuing that.
Yeah. We're already doing that, right? And then another thing that we're doing is, the algal, because the algal system has very high protein, and then combined with the carbohydrate, now we can do fermentation on that because it has already got nitrogen and it's like a rich media. So
Correct.
we are trying to see whether we can do something like a food fermentation on that and try to get, the flavor and the things right. So that it could be like an alternative, like, for example, plant-based cheese or something of that kind, where the protein from the algal system, right, on that you grow the fungi or you ferment that, right? So those possibilities are there, and we are already attempting some of those here.
Yeah, very excited to see how that goes, and I can't wait for the first sampling of that. So moving on, there were a couple of questions about gaseous fermentation in India, and I answered that by saying yes, we do have. Like I think Professor Venkatesh also mentioned that in his chat that he helped a methane-based fermentation bioprocess optimization for a company called String Bio. Funny enough, the coincidence here is literally an hour and a half ago, I was at String Bio's headquarters here in Bangalore, visiting them, and they do some brilliant work converting methane into single cell protein sources. And the update here is that they are not only doing it for feed now, they also have products for human food applications and cosmetics and so on. So they're really growing very well at scale. So a company to watch out for in the gas fermentation space.
Yeah. So the fermenter design, can be optimized where the gas distribution and all that, the uptake of those gas are possible. So that is very much possible.
Yeah. So if you have any questions related to that, please feel free to reach out to us, and we'd be happy to connect you with Professor Venkatesh. And thank you so much for sharing your thoughts and being ready to mentor them in that space.
Yeah. Anybody wants any help and all that, they can always approach, including facilities at IIT Bombay.
Thank you. Yeah. So moving on, there's a question on, can photobioreactor use sunlight as a light source? I think that's Professor Venkatesh, you were talking about photobioreactors for your algal system.
Yeah. At one level, optimizing the light source for a particular metabolism to occur, right? So that itself is one of the inputs which has to be controlled. And, by making sunlight as a light source, again, you lose that control, all right? So the right frequency and all that. So a photobioreactor is done so that you can regulate the light source, to the way that you want, right? To the regular hertz that you want and the complete control of the light source. So the patent that we have is actually trying to get that distribution of the right light, right? And not the sunlight, because sunlight again is going to get diffused, and then you're not going to get that optimal. The very point of photobioreactor is that you want to have a controlled light.
Correct.
There are some commodity fermentations that use photobioreactors, but a lot of these come with challenges with heat and contamination as well. So for food, it's probably not the right thing to do. There are a few companies trying to innovate in photobioreactor design with like fiber optic transfer of light and then filtration. So there's a company in Israel doing this, but it's complex. So I think really trying to have a controlled system, that's one of the tenets of fermentation. You want to have the microorganism in exactly the same state that you want it to be at all times.
Yeah, exactly. And especially, there are some spirulina and those kinds of growth that people do, which are in open pond in sunlight. But then, they have their end goals are different, right? So they just want to produce spirulina and then put it out as one of the products. And so each time they also face a lot of problem because seasonal variations are there, and then there is a lot of variation, contamination issues are there. So that is a challenge if you try to put it into sunlight.
Correct. That's, thank you for those pointers. Very, very important to consider if you have an idea. Okay, so there is another question here about gaseous fermentation models. I would like to understand more about the mass transfer efficiencies and the yields in gas-based fermentation models using carbon feedstock. So I would open it up to both of you because you're both experts in the field.
Professor, would you like to go first, or?
Yeah, you can go ahead, Adam.
Yeah, I think this is a question that's really highly dependent on the bioprocess and bioreactor design, but also on the particular carbon feedstock. So there are going to be mass transfer efficiencies that can be quite high, especially if you're co-located near a source of methane or carbon monoxide in the case of some of these organisms. And really, bioreactor design is going to play a larger role in gas fermentation, I think, than in liquid submerged fermentation, where there's bubbling through. Because if you are just looking for oxygen transfer, we really understand that well. But looking for the transfer of oxygen plus methane or oxygen and CO2 or CO, these are really complicated. And I think the Wood-Langdal cycle sort of methanogenic bacteria, we don't know necessarily as much about them as we should. So we're learning, and I think there's a wide open space here. This is something that is proving relatively difficult to scale, but I think with time, we'll either figure out how to scale it up or scale it out.
Got it. And the difference there between scale up and scale out is building smaller multiple reactors at one scale rather than going bigger.
There may be potential for that, and there may be some organisms that just tolerate that much better given the changes in mass transfer or pressures that can happen at large tanks. So the key would be to make them more cheaply than we do now.
Got it.
Yeah, there is a company in Denmark which has a patented bioreactor design for gaseous fermentations, right? And then I forget the name of the company now because we sort of contacted them as well. so they have scaled the distributions. They do a lot of CFD simulations and then try to optimize the gas flows and then trying to optimize, try to get the right mass transfer and the right distribution. So all those techniques, engineering techniques, will obviously be useful, in scaling up such fermentations.
Got it. And Adam, can you recall the name by any chance?
I was trying to think of it as well, and I can't remember. The great thing is there's so many companies in these spaces that sometimes it's hard to recall who's working on what.
Yeah.
Got it. I'm sure we can Google it. It's fine. But okay, so moving on, there's a question on how can we revolutionize the dairy industry through casein and whey protein through precision fermentation? What are the current white spaces and challenges faced by India in this sector? So I think Adam covered like a portion of this, talking about how these sort of techs tend to just scale in no time once it's recognized, and now is the era for precision fermentation derived dairy proteins. I love how Adam covered that portion. About challenges faced by India in this sector, I would say there are companies already doing this. There are 2 or 3 startups that are already working on creating whey, casein, and even lactoferrin. In fact, they are really taking off. I think immediately the scale up and the availability of facilities is something that they're still looking into. And of course, the regulatory side of things in India has not gained clarity yet. So they are the novel food regulations are yet to be framed by the FSSAI. So that is one challenge, I would say, currently for the precision fermentation derived dairy industry. And GFI India is working closely with the Indian regulators to try and make that more transparent, so these companies have a clearer path to market. Because as of now, they are in their R&D stages, they are ready to scale, they know what they have to do next. But given that they are producing a food ingredient, it would be better for them to know what the regulations are surrounding the inputs that go into making these dairy proteins. So that is one challenge that just obviously pops in my head. But about current white spaces, Adam, Professor Venkatesh, can you think of any white spaces that are particularly prominent in Indian sectors?
I'm not very sure, but as you said, right? So there are companies which are doing that, and they are in that optimization stage and scale up stage. And then most of them, are with no clarity on the food, are actually trying to go into the cosmetics and those applications first, right? In some of these products that they're producing, so that they have some footing and at least get that purity. And then that level of manufacturing is what they're trying to do. But actually, yeah, I mean, some inputs to them will obviously be helpful. Many of them have some challenges in terms of scaling up, in terms of getting stable strains, right? So they do have these challenges, and I think support at that end will surely be helpful to them.
And Adam, do you have anything to add?
Yeah, I agree. These are sort of the ways to increase efficiency and lower cost are going to be global challenges for everybody. And I think we are starting to see folks center around particular platforms for growth, but we'll see some innovations that potentially different chassis organisms or new downstream processing technologies are just going to drive that cost down enough to be cost competitive with animal-derived proteins, which is their main challenger.
Yeah. But there is hope in India, at least for, given our expertise in the biomanufacturing sector. I think if we really put our efforts now and take it up, we can address these challenges. We are uniquely positioned in that way. And in fact, Perfect Day, which is a US-based milk protein maker from precision fermentation, they've actually managed to get their FSSAI Indian regulatory approval to manufacture their proteins and commercialize it in India. So that is a huge green light for the sector itself. And hopefully they start production here soon, and that kicks off the industry here. So that's another way we can see the bright side of things. So just a couple more questions and we'll close in a few minutes. I think Shiryan is talking about, he's added a follow-up here about this is in context of acetogens using carbon. Oh, this is for the gas fermentation and mass transfer question. Do you have anything else to add in that context?
There are a few innovators, I think, upstream in trying to do things like potentially generate acetate that might change some of this, where there may not be actually a need for the gaseous inputs for some of these acetogens. There's a great paper that came out last year that talked about potentially having an electrical carbon cycle, where we can use electricity to take carbon dioxide and take water, and then we can make lots and lots of acetate and acetic acid, especially if you can get clean energy. And then this could be a potential feedstock where you could feed this to some plants for non-photosynthetic growth and non-photosynthetic carbon, but you can also feed it to microbes. So I think thinking even further upstream than the fermentation itself, but thinking into how we generate these feedstocks, this would be sort of an incredible sort of dual use technology where we could sequester some carbon, but then also use that carbon to build it into feedstocks. So I think the paper was in PNAS last year, but I'm again blanking on the name. I apologize.
That's okay. We managed to find it, and I'm pretty sure you may have updated this on the GFI resources page about the science of fermentation. So thank you for sharing that. Yeah, I'm pretty sure it's there because
I think the different feedstocks, right? So like acetate, what Adam was indicating, glycerol. So all these are other alternatives which that people are looking at instead of just the routine, glucose and the other factors and gaseous one adding their own challenge. So there are other alternates that people are looking at.
Yeah, great. And there is a subsection in the Science of Fermentation page exclusively on diversifying feedstock for fermentation. So that is a great place for finding more up-to-date resources, like Adam just mentioned, papers that come out. All that is usually very up-to-date on our webpage. So please make use of those resources, and hopefully they are continuously answering your up-and-coming questions. And the last one here is about heterotrophic cultivation of algae. Can it have more nutritional content? Sohail asks, because Euglena algae can grow heterotrophically. Any thoughts here, either of you, Professor Venkatesh, Adam?
So each organism has its own unique, advantages that you have, correct? And so until unless we actually see what is their productivity, and so they may have nutritional content, but they may have other challenges, correct?
Yeah.
So we need to look at a holistic way, and then it's always a challenge which has to be solved, right? And if it makes sense, economic sense, and then it can be scaled, and so obviously that would be one of the choice of the organism, correct? Yeah. So for example, we started working on mushroom. We saw that there are so many very, very specific compounds that they produce, and it gives that umami flavors and taste, right? So which again meat eaters enjoy. So bringing these things together, correct, with the algal fermentation and these right, so gives an opportunity. So similarly, I'm sure there will be specific opportunities that are there if things come together. So we need to take it as an opportunity and then see, right?
Exactly.
whether that makes sense. Correct?
Yeah, and you gave a brilliant overview of what are the different parameters to consider in your presentation when you're evaluating strains or the secreted proteins and other biomolecules. So I think that was a great masterclass on both biomass fermentation and precision fermentation derived protein for application in all proteins. So thank you so much, both of you, Professor Venkatesh and Adam, for and we've gone 15 minutes over time. Sorry about that, but this has been a very informative session. Can't thank you both enough for participating. Thank you. Hope to have you soon on a similar webinar style discussion, because it's so educational even for me. So thank you.
Thank you for hosting.
Thanks for organizing this, and then a pleasure to be part of it. Enjoyed being part of it.
Thank you. That means a lot. Thank
Bye.
you. Bye, Adam.
Thank you. Bye.